about Oscar. This will not happen now, but it will happen on Wednesday, tomorrow. Yes, tomorrow is the Oscar session at 6 p.m. European summer time, so make sure not to miss that. Today I'm talking about open loop and closed loop brain stimulation procedures. I just have to move the Zoom over there to see the full screen. Yes, and yes, great. And especially optimizing those procedures. So I have separated the talk into 3 parts. The first part will be more technical. It will be about hardware and software solutions we have developed here at g.tec. The second part will more focus on clinical procedures, more established ones. And the third part will tackle the latest novelties we have come up with. So this is the slide I usually start with. You can see a complete overview of our multipurpose brain stimulation research setup. As you can see, it consists of 3 devices manufactured by g.tec. It's, of course, a stimulator because stimulation needs a stimulator. Here, the G.Stim Pro cortical stimulator. And we have the G.Amp, the biosignal amplifier, which is recording the signals. And in between, we have the switching unit, which kind of connects those 2 devices. So conceptually, with this setup, we are able to stimulate, and we are able to sense, and we are able to switch. So basically, the cortical stimulator is connected to the switching unit with the stimulation cables, plus and minus. And the switching unit is able to programmatically route the stimulation channels to dedicated electrodes. So there is no more need to plug in and plug out electrode cables, but it's possible to do this entirely in software. And at the same time, we have usually many, many electrodes connected. At the same time, we are able to record the brain signals from all the other electrodes. They're also connected to the switching unit, and from the switching unit, they go into the G.Amp. And the G.Amp is then digitizing them and forwarding them to the control PC. Notably, it's not possible to record the stimulation channels at the very time of stimulation, but it's possible to record them immediately before and immediately after stimulation. And by immediately, I'm talking about milliseconds. So depending on the setup, 4 to 6 milliseconds we're talking about here. It was already mentioned open and closed loop, so it's very important to make this difference here. Open loop means that before you're starting the experiment, you're setting up all the stimulation parameter and the whole stimulation protocol a priori, and you're not really reacting to the response of the patient or to the recorded brain signals or biomarkers you're extracting. So when you want to do that, when you want to react, and especially in real time to the response of the patient, you're talking about a closed loop system. So here, for example, you would take the recorded signals from the G.Amp, you would extract some biomarker you're looking for, and then you're using this biomarker to generate some control signals. You're then feeding into the stimulator or into the switching unit. So this is closing the loop, and therefore it's called closed loop system. We can use 2 kinds of different trigger signals to initiate a switching or a stimulation procedure. So one thing is just doing this by stimulation commands. So just giving them an API call via the software. Or if you want to go faster, you can also use hardware triggers. So this means they are physically connect with a cable. For example, the stimulator and the switching unit, they have a physical connection via a trigger cable. And depending on who is triggering whom, so either the stimulator, the switching unit, or vice versa, one of those devices sends out a TTL pulse, so a 5-volt pulse, which then immediately issues the other device to react. So this is very important to know. Since we are connected to the control PC at all times, we are also able to change those settings we are using. So we are able to use adaptive settings for stimulation. So we can change the amplitude, we can change the pulse rate, we can change the pulse width and all that stuff. in real time during the experiment. But of course, we can also change the stimulation channels or the group of stimulation channels we are routing the stimulation current to, all that in real time. Exactly, and with the HIME, we are able to do all this processing in real time, and with the g.tec software platform, which is a Simulink toolbox, we are able to very quickly prototype new neuromodulation or brain stimulation experiments, because HICIS just features building blocks for the stimulator, for the switching unit, and for the high amp, and you can just connect them and set up your experiment. So since the switching unit is kind of the most recent device, I also have a list of core features, but I mainly explained this already. Importantly, you can not only route the stimulation to pairs of stimulation electrodes, but you can also group electrodes. So for example, you can select electrode 1 to 16 and just route the stimulation input into those electrodes, and here the current then distributes according to the electrode impedance. So you can just group them, and the same goes for the negative stimulation polarity as well. What is also very nice is the built-in impedance measurement. I will demonstrate just in a couple of minutes, and we also have a graphical user interface to control the switching unit. If you want to go more into the research domain, as I said, we have also hardware triggers to speed up experiments and select a dedicated high-speed switching mode, which allows you to switch across different channels at the rate of 100 Hertz. So this is 10 milliseconds for each stimulation location. So this is also called the well, this is done in standard settings, and there is also an even faster mode. It's called fast mode, where we are stripping away the disconnection from the amplifier. So in this mode, the switching unit remains always connected to the amplifier at all times. So this saves a little bit of time. So this allows us to speed up the channel switching up to 200 Hertz, so a factor of 2. Yeah, then you can explicitly set your ground channel if you want to kind of modify your setup. We also have an audio out channel where you can route any dedicated electrode to an audio output, which you can connect here to the rear side of the switching unit. And I already talked about Simulink. So we have a Simulink block for closed loop integration, and also a MATLAB API to control the switching unit. So right now we have the switching unit available in 3 variants, 64 channels. So this is just the left group here with 1 electrode connector box, 128 channels. So there you have 2 connector boxes, or the full-fledged switching unit with 256 channels, which uses 4 electrode connector boxes. We're compliant to the most relevant norms, so medical electrical equipment, basic safety, stimulator norm, and EEG electroencephalographs norm. And very interestingly, or very importantly, quite recently we got medical certification for the switching unit as part of the CORTIQ 2.0, which Christoph Cabella has been presenting and will present in other talks during the spring school. Good, so this is about the hard facts about the hardware. But now let's look a little bit into the software. So here you can see a screenshot, but I will just briefly demonstrate this. Here, because I have the switching unit connected. I used the G Suite to start the switching unit software. I can now perform a self-test, but I have done it already. I don't need it. Exactly, and now here I have the control software. Looks like it's integrated into the slides. And I can just, select channels, whatever I want to stimulate. With the control key, I can toggle between positive and negative electrodes. And then I can just click activate switching unit. And now the active LED is on, and the switching unit has just routed its stimulation input to those dedicated electrodes. Good, so now this is just for demonstration purposes, and there's no stimulation going on. But what this app also features, and it's quite nice, is an impedance check. So I can record or measure the impedance of the electrodes in real time. So this gives a very, very important information about how the electrodes are connected, because you don't want and you cannot stimulate into electrodes which are not connected at all, because you're not able to put the current through the leads if they're not connected. And also you don't want to stimulate into electrodes which are connected, which are implanted, but which are not properly connected. So they're floating in some liquid or something. Because when the impedance gets high, and in this case, the impedance gets high, then the stimulation current will cause the voltage to increase, and such increased voltage will cause pain to the patient. So it's always important to keep the impedance low as much as possible. And here you can see, since nothing is connected, I have high impedance all over the place. But now I will connect, I can show you. I have a saline solution here and then a grid. So this is kind of salt water. It's mimicking the milieu of like brain tissue. So I put this into the saline and I will connect the electrode head box to the first group. And you will see whenever the stimulation impedance impedance measurement arrives at the first group, then hopefully the impedance will turn low for those channels which are good. Exactly. So here we have very nice low values for the impedance, except for a couple of channels which are obviously bad. So this is very important, because now I know that I cannot stimulate channel 14 and 23 because for some reason they are either broken or they are not connected properly to the patient's brain surface. All right, so let's stop the impedance check now. I also have an impedance check, which is a bit more versatile. So you can even close it now. With the research software, you can start the impedance measurement tool, which allows you to measure the impedance across frequencies. So this really gives you a nice feeling of the connectivity of the electrodes and also the characteristics of the electrodes with increasing frequency, because impedance per definition is the resistance of the electrode at a certain frequency. So when I start this impedance measurement with just standard settings, it's measuring the first 16 channels, impedance against ground, from 20 hertz to 1 kilohertz. And you can see that here the values I've seen in the previous GUI, they're nicely replicated. So here the stimulation was measured at 1 kilohertz. We get the value of about 1 kilo ohm, and as the measurement frequency decreases, the stimulation, the electrode impedance increases. So here we have between 10 and 20 kilo ohms at 20 hertz. This is also quite plausible and would be a good connectivity to perform recording and stimulation experiments. All right, so there is more to this GUI as well. If you want to identify which channels exactly are bad, you can switch to the channel view, and down there you have a slider which presents you which allows you to switch the frequency, and you can see channel 14, the same as we have seen previously in the GUI, has high impedance all over the place. So this is a channel which is not properly connected. All right, so this GUI also features export to figures, which are ready to print and to publish. So this is kind of a nice tool if you want to investigate the characteristics of your electrodes. All right, now let me continue with the slides. I already mentioned the Simulink integration and the g.tec platform. So here we have a Simulink block, which provides you with a real-time control of the switching unit during neuromodulation or brain stimulation experiments. So you can adjust the stimulation channels, you can adjust the ground channels, you can enable or disable the hardware trigger, you can enable or disable the fast mode, you can set the audio out channel, and you can just start or stop the switching procedure. So this gives you a very versatile control of the switching unit. And at the output, you have just feedback. You have an indicator in which state the switching unit is. So whether it's in ready state, so where nothing is switched, or it's in active state, where the channels are switched. And you also get a couple of arrows to nail it down whenever something has happened erroneously. Good, and then the MATLAB API provides you with a couple of features just to really program your switching unit in a non-Simulink environment, but in a MATLAB script, which is probably attached to some other experimental environment. Here it's important to mention that this is quite a fast API. So we have measured, and most calls are completed within 30 milliseconds. So for software control, this is quite okay. Yeah, I've shown you already the impedance check. Here again, maybe the specs. So we can measure the impedance from 1 kilo ohm to 1 mega ohm. So this is quite a wide range. So you can measure depth electrodes, which traditionally have very low impedances because they have large surface compared to maybe needle electrodes. Here you can see the blue curve is a needle electrode. They have a very small surface, so the impedance is very high. This is necessary. So this makes it necessary to have a rather large range for the impedance to be measured. And the frequency you can sweep from 20 hertz to 1 kilohertz. Good. Yeah, and the accuracy is also quite nice. We have plus minus 500 ohms or plus minus 10%, depending on what is greater. So the results you get are usually quite reliable. Good. So that much about tech specs and our equipment. Now let's move over to the brain stimulation procedures, the actual clinical and the research applications. So we have heard already previously a lot about the treatment of disorders by the amazing talk by Julian. That was really amazing to watch. But also we have the identification of brain functions. It's also called functional mapping. We've also heard about this by Christoph Capella and other colleagues. So these are the main domains, you would say, the main application fields of open and closed loop brain stimulation in the clinical field. And when we're talking about functional mapping, the most prominent one is probably ECS, electrical cortical stimulation, but we've also heard about CSEPs, cortical evoked potentials, and also SEP, somatosensory evoked potentials, which allows you to, for example, identify the central sulcus. Here, the sulcus between motor and sensory areas in the brain by stimulating the median nerve here. Yeah, and those quite established clinical procedures, they of course lead to, or they motivate and drive clinical research for more advanced protocols. So over the next couple of slides, I will go through the kind of exemplary applications of those applications, starting with ECS. You might have heard about it already, but let me briefly recap it. So when you're injecting current into the brain, you're overriding the natural physiology of the brain, and this yields either the excitation of a certain function, so you can make the hand move, for example, or cramp when you're stimulating the motor area, or it yields the inhibition of a particular function. So for example, when you stimulate into speech production areas, Broca's area or mouth area, while a patient is speaking, you would be able to just inhibit or block this function. So when you're doing ECS in a clinical setting, ECS functional mapping, usually you are stimulating pairwise the regions of interest where you suspect the function to be. And then based on the behavioral response of the patient, you can create your functional map. So de facto, this is the clinical standard since almost 100 years. And it's possible to do this not only at the patient's bedside with implanted electrodes, but also intraoperatively while the patient is under surgery. So typically, functions we can map with ECS are motor functions, so everything that moves basically, or somatosensory functions, so everything where you feel something, or also cognitive functions. So for example, spontaneous speech, picture naming, comprehension, language comprehension, but also calculation. So usually when you're doing ECS, you're using a biphasic rectangular pulse with a duration of 300 to 500 microseconds and an amplitude of 2 to 50 milliamps. And you are applying this for a couple of seconds at the rate of 50 to 60 hertz. So typically, when you're at a certain location, you wants to map, or you want to elicit a certain inhibition or excitation. You start with a very low current because you have to, safety first. You don't want to harm the patient or cause any discomfort. So starting with 1 milliamp, usually you don't have any response, and then you gradually increase the stimulation current in steps of 1 to 2 milliamps, and then you observe the symptoms. So at one point, at one certain stimulation level, usually you get some response, some effect. Or if you don't, if you don't observe anything, you have to stop at a certain level. Might be 10 milliamps or 12. And then, you proceed. You change the simulation site as needed. And of course, obviously, you have to very carefully monitor also the brain waves during the ECS session, because it can happen, especially when you're treating an epilepsy patient. It can happen that the stimulation induces so-called after discharges. So this is kind of a pathological waveform, which is triggered by ECS and can lead to a generalized seizure, which is something you want to avoid at all costs. So you have to monitor those after discharges, after stimulation. So usually when you observe them, those patterns, you just wait because usually they diminish after 20 or 30 seconds. So here's an example done by Professor Kamada, Dr. Kamada in Japan, where we see the left hemisphere of a patient with implanted electrodes. So the black dots are electrodes. And here at the motor cortex, we stimulate into the hand region with 4 milliamps. So the patient was moving the hand. And then when the stimulation happens, the patient is not able to move the hand anymore. Yeah, so this is a nice example. The patient was moving the hand, then the motor area was stimulated, and then the movement was not possible anymore because the hand cramped. Another example, which was more research related and is published in a nice paper by Gavin Schalk and Christoph Kopeller and other researchers, was the stimulation of the fusiform gyrus, which is responsible for kind of shape and color and face recognition during visual processing. So here you can see the view of the temporal base of the brain, kind of from below the bottom, and implanted electrodes. And here, when stimulating those electrodes, those are kind of face selective, is a face selective region. You will see that during the experiment, I will skip a little bit. During the experiment, when the patient was watching, for example, a black box with some structure, during the stimulation, the patient got some face-related sensations. So here, the box turned into kind of into a face, and also the ball turned into a face when stimulating. And And here, looking at the neurosurgeon Dr. Ogawa, the face completely changed during stimulation. So this is a nice example how you can also use ECS based on a clinical procedure, also for research purposes, because in this paper, the ECS was related to high gamma activity in this region, and quite nice findings turned out to come up and were then published. So in a very same setting, the color specific region was stimulated just a couple of electrodes away with the very same protocol. And here the patient reported that this structured black box turned into a very colorful object, I think rainbow colors. Okay, and the same then for the ball. And so on. I think you get the point, right? So this is a very, very powerful procedure, but of course you have to apply it very carefully. Good. Now moving over to CEPs. We have also heard about them today already. So basically, they allow us to reveal physiological networks and paths in the brain, but also kind of pathological networks. So when you're stimulating one side with the typical CCP settings, and you observe the response in all the other electrodes at different cortical locations, and you are averaging the responses as evoked potentials, you can see the typical CCEP shapes at locations which are functionally connected to the stimulation site. So this is very nice because it allows you to reveal brain networks just with one stimulation. And also it can be done without the patient's involvement. So this the patient usually has no sensation. You can do it just basically passively without any interaction with the patient. So in this example, when you stimulate into Broca's area, which is about here, then the mouth motor area was nicely revealed about here. And also at the same time, the Wernicke area, which is the receptive language area, was also nicely revealed here. So we put this into an autonomous screening experiment using Simulink, G.HYSYS, the switching unit, high amp, and the stimulator, so our brain stimulation setup. And so this is fast forward playback. You can see the stimulation sites here, plus and minus. They are switching after a couple of stimulations. And here in the middle, you can see the evoked potentials caused by the stimulation. And the top right shows you the amplitude of the CCP responses as bubble sizes. So I think Christoph has shown this today already, but I will go to just 1 minute and 5, because this gives you a nice response here. Oh, let me see. Yep, here. So here, the similar as in the previous slide, stimulating into kind of Broca's area here, speech production, elicited very big CCPs at the mouth motor area. So here, those channels. This was a very nice nice outcome, but of course, you know, during the CCP screening, lots of locations are stimulated, and this overall then allows you to create the functional map of connectivity regions of the brain. All right, so this is all open loop. So this is just, you know, a predefined protocol which is executed. So now let's move to a typical closed loop example. So Julian was also brilliantly explaining this in much more detail, but I just want to mention it here because it's one of the predicate kind of examples for a closed loop brain stimulation. It's adaptive deep brain stimulation, which is used to treat patients suffering from Parkinson's disease, for example. So here it has turned out that when you are measuring the brain signals in subthalamic regions, and when you're extracting the beta power, this can be used as a biomarker, as a control signal to initiate brain stimulation, to make the overall deep brain stimulation setup much more effective. So the typical procedure would be to record the local field potential here in the subthalamic regions. You are filtering to the beta frequency range and compute the power or the amplitude of this signal. And then based on a threshold, using some statistical evaluation, every time this threshold is exceeded, you trigger the stimulation into the deep brain areas. So this is the schematic procedure. And one research setup using Simulink, you can see here. I'm not going to talk too much about the details because I have other experiments coming up, but I just want to briefly hop to second 50 because here you nicely see how that looks. Here in this scope, you see the beta power and the threshold in red in real time. And here you can see the GSTIM Pro control software, which is just waiting for the trigger signal, which is derived from this threshold to be exceeded. So this is now played back a little bit slower to keep track. But now you will see that the power overshoots, and immediately when this threshold is exceeded, you have to watch this here, the stimulation is initiated, okay? Right, so here sequence running means stimulator is active. Good, so this is a typical example for a closed loop stimulation. Right, good. Now let me come to the third part of my talk. It's about novel neuromodulation features. So the previous years, we have been working a lot on the switching unit, but in the last year, we have more worked on the stimulator side. So we have come up with a couple of new features for the stimulator. And the first one being to widen the range of pulse shapes we can deliver. So the GSTIM Pro already is able to deliver quite stimulation pulses with many configuration parameters like amplitude, width, pulse rate, train length, and so on. But people have been asking to deliver more alternative pulse shapes which are not perfectly symmetric. So symmetry in one way is very important because when you are symmetry in a way that the positive and the negative lobe of the pulse have to be inject and take back the same amount of charges because you want to keep the charge level at the same you want to keep the charges at the same level. But it's possible, for example, to change the timing of such a stimulation pulse, where the first part is shorter and higher in amplitude, and the second part is longer but lower in amplitude. So there are a couple of research applications which want to investigate this. So the charges which are quickly injected into the brain in the first point are only slowly drawn back by the negative lobe here. So this is how it looks when we record the stimulation signal with the amplifier. And notably, this is so fast that the shape here looks a bit like the transients don't look so steep, but this is only due to the sampling rate of the amplifier. So the stimulation signal itself, when you look at it with an oscilloscope, is perfectly sharp. So it's also possible to modulate amplitudes now more flexibly. For example, you can ramp up stimulation amplitudes here in a sawtooth pattern, and you can go up and down, causing a triangular pattern. You can also use a gated sinusoid to create these kinds of patterns, or you can also even randomize in case you want to inject a random stimulation at some point into the brain. So these are the new advanced pulse shapes we have come up with. And some other requests have more focused on the timing to get even faster when configuring the stimulator. So here we were able to make the stimulation configuration much faster. So previously we have been at 500 milliseconds, but we've improved the firmware and software. So we are able to update the stimulation parameters at a rate of 80 milliseconds now. And when it comes to initiating stimulation, previously we had a delay of 100 milliseconds just from software command to actual start of stimulation of roughly 100 milliseconds. But we improved the software trigger delay to only 22 milliseconds, which is also about 4 to 5 times faster and also more accurate because the jitter here is smaller. So for those who are not yet satisfied with the 22 milliseconds, we've come up with an Arduino control. So this is a very small microcontroller. You can connect to the USB and you can communicate with this nice board to generate an actual hardware trigger for the stimulator or for the switching unit. So here we were able to get the 22 milliseconds down to 5 milliseconds. So this is again a huge leap in trigger onset delay. All right, so now this all put all together into like advanced clinical research setups. We have done two, or like today I have as an example, I have two setups I would like to present you. One of them is very nice because it's very visual. It's about high precision visual stimulation. So here we can here we link electrical stimulation to visual presentation of stimuli. So we have 4 conditions. One condition is that there is no visual stimulus, no picture shown, and no stimulation, no electrical stimulation. One condition is that there is a picture shown, so visual presentation. One condition is we have a picture and electrical stimulation. And the fourth condition is that we have only electrical stimulation. So this, for example, could investigate how stimulation, like during a picture naming task, how stimulation affects the face, or how stimulation right before a picture is shown, or while a picture is shown, or after a picture is shown, affects the overall behavior of the patient. So these stimuli, they have to be shown on a monitor, of course, and all this has to be set up in a so-called paradigm in an experimental protocol, in a procedure, how they are shown on the screen. And this paradigm is usually created with Paradigm Presenter and then just played back on the monitor. So when we want to stimulate and to switch, we need the GST Pro and the switching unit, which we configure. And then we need trigger signals. So here we use, because we need very high precision triggers, we use photodiodes. So one photodiode, the orange or brown one, for the stimulation trigger. So this is why you see those white rectangles here. So you put the photodiodes directly where those white rectangles appear. So every time your condition, the stimulation condition, is active, the photodiode reacts because there is a contrast change from black to white. And the trick box converts this photodiode sensed signal into a valid trigger pulse, which is then used to issue the switching procedure of the switching unit. And then the switching unit forwards this trigger signal to the stimulator, right? once that the stimulator receives the trigger signal, it immediately issues a stimulation through the switching unit into the electrodes at the dedicated locations. And at the same time, we are recording the brain signals with the high amp. So we still need to properly analyze the experiment. We still need the trigger signals also recorded by the high amp. So for this purpose, we use the picture trigger captured by the photodiodes. We use the stimulation trigger, and we use the output trigger signal by the stimulator, which really identifies the time point where the stimulation has happened. Put this into the digital input of the high amp, which synchronizes the digital input with the recorded brain signals, and then forwards it to the control PC where we can analyze it. So step by step, I think this is quite intuitive, and there have been research groups which are performing exactly these kinds of research procedures. So the second one, if we have still time, yes. The second experiment I want to show today is also involving high gamma activity. So we have already heard about high gamma activity. It's a very robust biomarker of behavior or cognition. So when you're doing something, when you're moving, the involved cortical region shows a power increase in frequencies above 50 hertz. So you can perfectly well capture this with ECOG electrodes, and you can just extract the power of this frequency range and use this as a control signal for various BCIs. So the question here is if we can use high gamma activity to optimize ECS and also to reduce the risks. So yes, high gamma activity can be used to reduce ECS to the relevant locations. So this is exactly what CortiQ 2.0 is doing. So before we are going into the ECS functional mapping, we're doing high gamma activity mapping, and then we already know very nicely where the hotspots are, and then we don't have to stimulate too many regions because we already know the relevant ones. And the second question was if we can use high gamma activity to find optimal stimulation timing. So this has been so far an open question, and up to some degree it is still. But together with Dr. Kamada, we performed a neuromodulation study where we wanted to tackle this question. So in this neuromodulation study, we had two patients doing an expressive language mapping task, so a picture naming task. And during the phase of where the picture was shown and the speech and was named, randomly we injected ECS stimulation with a random timing into the speech production area, which was mapped beforehand. And then close to the stimulation site, we extracted high gamma activity for analysis purposes, because then we analyzed the response of the patient and related the speech onset to the timing of the stimulation and to the high gamma activity time course. So to give you an impression here, this is already submitted publication, so let's hope it gets published soon. Here you can see a standard picture naming task where the patient was seen a couple of eggs, and in real time you can see the high gamma activity on the left hemisphere. So this is a very short clip, but make sure to pay attention to these areas. So they will highlight first, there will bubbles appear because this is the visual cortex. So this is what happens first. So there's a visual impression, and then the speech production area gets active here. So I will play it back now. Right. So as I said, first of all, the bubbles appear here, and then the patient was pretty fast. Here, the speech production area gets active here. And you can see it also here on the in a spectrogram. Right after, I think 3 milliseconds after the stimulation, you can see nicely red areas here. So these are power increases, highly significant power increases above 50 Hertz, right at the speech production site. And here you can see the typical or the recorded speech onset for a couple of runs, trials, with the audio waveform shown here. So this is now without any stimulation, right? So this was just showing pictures and recording the high gamma activity and also recording and noting down the speech onset. So this is also called the sham run, no stimulation at all. So when it comes to stimulation, it's here we have a very nice example for the same stimulus. So this was also X. So definitely the patient was able to speak out very quickly the like tamago, the Japanese word for X. But when we stimulate this area, you will see that during stimulation, she's not able to speak out. So she just speaks after the stimulation. So I will play it back now. Right, so it takes maybe 1 second. And afterwards, she's able to speak. So clearly we saw an ECS effect now in this very single trial. So there are a couple of trials, lots of trials recorded. And you see an overview here. So these are the trials with stimulation. So in yellow, you can see the window, the time frame of the electrical stimulation. And the black dots indicate the speech onset. So you can see that they tend to be more right? So right is slower, later. So stimulation clearly has an effect, but this is nothing we can quantify quite easily. So in the next step, the question was, how can we extract or quantify the impact of ECS on such speech production tasks? So this is the main result. Maybe starting with the figures on the right. So these are the two patients. On top, you see the high gamma activity time course, SC scores over time, with a certain onset, with a certain peak, and kind of a decay indicator. So what was interesting here is that, and this is the main observation, if you are starting the ECS stimulation before the high gamma activity peak, you have much more effect with ECS on the behavior as if you do it afterwards. So this is the relation between the black and white dots here. So when you start before a high gamma activity peak with a simulation, you can see many black dots, right? And black dot means that we have an ECS effect, meaning that there is no response at all, or there is a speech disturbance, notably. So 92% here of the experiments with ECS before the high gamma peak showed an ECS effect, but only 37% showed an ECS effect if you stimulate after the peak. And also very nicely visible is that the white dots, they are more like on the down there on the bottom. And this means that the response is faster, because on the y-axis, vertical axis, you see the response onset in milliseconds. So as the dots go up, the response is later. So this is also quite intuitive. So for patient 2, it's the very same observation. If we stimulate before high gamma activity peak, then 47% of the trials are affected by ECS. And if we stimulate afterwards, only 13% are affected. So this is exactly shown by these tables. And you can also see this in terms of the mean response onset. So if there is no ECS at all, you see that the speech onset is faster than if ECS is applied, whether it's after the peak or before the peak, doesn't matter. And especially comparing the sham experiments without ECS to the ECS before high gamma activity peak, there is a strong significant difference. Good. So that's basically everything from my side. Thank you very much for your attention, and looking forward to your questions.
Thank you very much for the nice demo. Chandra is asking for the deep brain stimulation. What is the time difference between the sensing versus the stimulation?
It really depends whether you use hardware triggers or software triggers. But usually it's a couple of milliseconds. So when you're using hardware triggers, you would be able to do this within 5 milliseconds, and with software triggers, you would be able to do this within maybe 20, 30 milliseconds.
Another question is, what is the price of a eStim Pro that's FDA cleared?
Here, I have to forward this question to the sales team.
It's about 18,000 euros, I guess. But who knows what Trump is doing in the US? Could change in the US anytime. Eleni is asking, did the high gamma timeline show earlier or more sustained activity in Wernicke's area? That might explain why ECS there didn't cause immediate disruption compared to Broca's area.
Ooh, good question. We haven't specifically looked into that because we analyzed high gamma activity right next to the stimulation site. But Wernicke is mostly speech reception, so I don't know if this would have a relevant information here. Also, I don't know if Wernicke was probably covered. Could have been possible. Yeah, it would be a nice site investigation. Thanks for the input.
So Joy is asking the time of the day, circadian phases and oscillations. Does this have any influence on closed loop experiments? So could there be a day of the time or a time of the day where the neuromodulation is more effective?
I think this is an open question. I don't know if this has been addressed, but I would say that there is no real effect except whether you're sleeping or not. I think this would have an effect, of course. But other than that, the circadian rhythms, I don't think that this has an influence.
When you're using the electrical cortical stimulator, where do you place the electrodes?
So those electrodes are implanted, and they're really based, so the location is based on the clinical needs, of course. So depending on for epilepsy patients, depending on where the epileptic focus is, or for tumor patients, depending where the tumor is located. So this is driven by the clinical needs. But we also had research partners who, for research purposes, inserted additional grids at interesting locations, especially motor areas, even though they are not always were not always relevant in those cases. But yeah, the good thing is with ECOG, with those grids, you have, especially with high density grids, you have a very, very high spatial resolution. By just inserting 1 or 2 grids, you already cover good parts of the motor cortex of the cortex at all.
Why is it important to measure the impedance of these electrodes, and what's the difference between standard electrodes and small electrodes, high density or micro electrodes?
So the measuring the impedance is important because you don't want to stimulate into areas that are not well connected. So when you have floating electrodes, for example, the impedance is high, which causes high stimulation voltages because the current is always the same you're injecting, and this causes pain to the patient. So it's one very important reason why you have to monitor the stimulation impedance. And another, especially for research, closed loop protocols, another reason is that the stimulator would interrupt stimulation when it cannot stimulate anymore. Because when you have an open lead, when an electrode is broken or not connected, the impedance is high or infinite, and you cannot stimulate anymore. So this means on the one hand, it's the patient's comfort, and on the other hand, it's the technical requirements of the setup, which just needs the impedance monitoring. And regarding the electrodes, so it really depends on the exposed surface. So depth electrodes with rather large exposed area, they have very, very low impedances, can go down less than 1 kilo ohm. And needle electrodes, they have a very, very small exposed surface. Here we can reach hundreds of kilo ohms.
So what would happen if you stimulate into an electrode where the impedance is too high?
So if the clinical threshold, the medical threshold is exceeded, so there are it's like safety mechanisms in the stimulator, then the stimulation is immediately interrupted. So the stimulation just won't take place. But even below this safety threshold, it can happen that the patient has huge discomfort or feels pain because the stimulation voltage gets just too high.
And it's also fascinating if you look actually with an oscilloscope at the current output of different stimulators that are existing, how much noise they are generating. So we were super surprised how noisy these signals are. And this makes also a difference how much you are stimulating certain brain areas and how much noise you are generating. Another question is, why do you call it impedance and not resistance?
Because impedance is per definition the resistance at a certain frequency. So impedance is frequency-dependent resistance.
So in EEG measurements, mostly impedance is measured at 10 Hertz because this is the alpha frequency hotspot. This is where you are interested in measuring good signals. What about ECOG? So which frequency are you choosing for these signals?
For recording, we use 20 Hertz usually. And 1 kilo for recording impedance. So there are kind of two impedance categories we have defined. One is for recording, because also when you record signals, you want to know the reliability of your signals, right? You could record signals which look good but are essentially only noise, and the impedance check just allows you to quantify the kind of connectivity of the electrode. So here we use 20 Hertz, just out of convention. And for stimulation, we use 1 kilohertz, just because many stimulation pulses have 1 millisecond pulse length, which corresponds to 1 kilohertz.
But Corteq also allows you to look at all the different frequency components, and you get actually a nice plot where the impedance is at different frequencies. And this is super important because you're not measuring just at the single frequency, of course. Then Arthur wants to know, is the hardware 100% developed and manufactured by g.tec, or do you work with other companies?
So the main components, the stimulator, switching unit, and high amp are entirely manufactured by g.tec. So the electrodes, for example, they are not our business. So we are ending at the electrode headbox level, and what you connect to the headbox is then a third party. But of course, we have a huge list of compatible electrodes we can record with. Yeah, but other than that, everything is entirely g.tec.
EP wants to know if a biomedical engineer can contribute a meaningful way to DBS surgeries.
Probably yes, if you find a lab where you can participate in studies, for example, or develop the procedures further, why not?
Actually, for sure, because neurosurgeons need biomedical engineers to do the technical aspects. So they are super busy delegating 20 people in the room and to insert electrodes. So there must be an engineer who is overseeing all the hardware and software components. So Johannes, this was very nice. Thank you very much.